Good evening, everyone. A very warm welcome to all of you present here. I'm Dr. Noori, Senior Editor at BHMS, and I'm truly delighted to be your host for this highly anticipated webinar brought to you by Homeopathy 360. Today, we are stepping into a fascinating topic, research frontiers in homeopathy through nanodynamics and advanced therapeutics. For over 2 centuries, homeopathy has inspired both curiosity and conviction, but in recent years, science has begun to uncover how deeply material and dynamic this system truly is. The field of nanodynamics is now redefining homeopathy not just as a holistic or energetic medicine, but as a scientifically verifiable nano quantum system. Today's discussion brings forward a colossal research spanning more than a decade, research that offers concrete scientific evidence for the very basis of Hahnemannian pharmacodynamics, uncovering the scientific proof of material content in ultra high dilutions, all brought together in one groundbreaking volume, Nanodynamics, published by BJAN. It's an absolute privilege to welcome our distinguished speaker and author, the visionary behind this pioneering research, Professor Dr. E.S. Rajendran. Sir, we welcome you. Dr. Rajendran is a name synonymous with scientific advancement in homeopathy. He is the first researcher to discover the presence of nanoparticles in homeopathic drug potencies, a revelation that has transformed our understanding of this field. Currently, Dr. Rajendran is engaged in a remarkable collaborative project at Rajiv Gandhi Center for Biotechnology, further bridging the gap between molecular genetics and homeopathic therapeutics. Beyond his laboratory achievements, Dr. Rajendran has been an eminent academic professor, examiner, and prolific author. With great pleasure and deep respect, I invite Professor Dr. E.S. Rajendran to share his insights on research frontiers in homeopathy through nanodynamics and advanced therapeutics. Sir, the floor is all yours, and we welcome you for taking time out for us today.
Friends, my dear doctors, my dear students, it's a great day, great evening. Thank you everyone for coming for this seminar. And thanks, Dr. Noori, for your great introduction.
Pleasure, sir.
Okay, you be there. So, today we are going to speak about research frontiers in homeopathy. Why we say that homeopathy is the most advanced medicine today available in the world. There are definite reasons to say that homeopathy is a highly advanced medicine. We have scientific evidence, we have clinical evidence, and we have evidence from all over the world where the practitioners of homeopathy who enjoyed the cure in homeopathy. And we are trying to find the reason why and how we are able to cure the patients. This is an institution where I'm rightly working. It's a tertiary care hospital in Pala, Kerala. And we have a complete Ayush department in that, and we have a strong homeopathy department also. And I work in the hospital after my relieving from the academic field in 2019. So thank you everyone for joining for this evening. So before I start my proper subject, I'm going to have another 2 more sessions on November 18th evening and November 20th evening. That is related to the textbook of Nucleus, Lectures on Chronic Diseases and Miasm, recently published, the second edition by BJ. And also New Lights, Lectures on Homeopathy and Philosophy, a book which was read by majority of postgraduate students in India from 2000 to 2025 period. So we are going to have 2 more sessions. So this is the first session today. And many of these books are being translated into different international languages like German, Spanish, Russian, Turkish, etc. So this is a book we are going to introduce today, that is Nanodynamics. The first edition of Nanodynamics was published in 2015, and the second edition was published in 2019. And this is what BJ has rightly published now. And I'm so thankful to BJ to take over this work from my burden, from my head, the burden has gone to the head of BJ. Thank you so much, BJ. And this is the German translation, which is available in German language right now, which is called Nanodynamic, that is in German language. So my study started in 2010. I was curious about why homeopathy is working and how homeopathy is working, and I was unhappy and unsatisfied with the philosophical explanation given to a science. Philosophical explanation always is the basis of every science, but at the same time, it needs to be connected to the science of cause-effect relationship. That is, we need to know exactly what is a process. So for that, any amount of philosophical explanation is not sufficient, and that we call human curiosity. And the human curiosity is what we all have. When we treat a patient and cure a patient, we are curious to know how the disease came and how the medicine acted and what was the process of cure which in the patient. This is what we call the whole science of medicine. So during my journey into the research in the last 15 years, the last 12, 13 years from 2010 onwards, I was exclusively working with modern electron microscopy and spectroscopy, and also select all the sort of dynamic light scattering. And my work was in nanotechnology department. And I used two nanotechnology department extensively for all this more than a decade time. One was the Indian Institute of Science, Bangalore, the nanotechnology department, and another was International and Inter-University Center for Nanoscience and Technology, MG University, Kottayam. And I was lucky to have a team of collaborators, professors. They supported me throughout my study. And these are the centers I worked extensively for 12 years. And for your understanding and to show the curiosity, I'm showing you the HRTEM and a high resolution transmission electron microscope attached to energy dispersive spectroscopy in MG University, Kottayam, which was one of the finest electron microscope, came to India in 2012 under the guidance of Professor Sabu Thomas. He was the director of the Department of Nanotechnology at that time. And they got some fund to buy this instrument, which was one of the instrument which gave me an intense ability to understand the depth of homeopathy in high resolution transmission electron microscopy. And definitely I was a researcher, an outside researcher, not an internal researcher of the university. So I was paying for every sample I was studying in the electron microscopy, but the time and the availability of facility which was provided by the university was of enormous help, because such an electron microscope was not available in, I think, in South India at that time. So this is what I am going to show you. In the Petri dish, you can see a triangular filter paper. On that there is a small rounded stuff you can see, and that is what we call TEM grid, TEM, TEM grid. And this is where this TEM grid is made up of carbon plate, and on that copper wires are fixed, and there are 200 columns in that. Each column is measuring 50 microns. And this is where we are putting our homeopathic dilutions after a process of 20 minutes of shaking. And that was what that vibrations which we give under the water. And that one we are using the dilution of homeopathic potencies in 30 ml bottles. We usually do a process called sonication. Sonication is means we put the sealed bottle and the water, and the ultrasound waves are passed for 20 minutes, and the sonication process give us, if there is any nanoparticles which get segregated. And this, from the middle of this bottle, one micro drop is taken by a micropipette and poured into this TEM grid. And the size is so small, and if there are particles of nanoscale, they get trapped into each column in the TEM grid. And there are 200 columns. And these columns, after 24 hours of infrared rays under that, because we have to dry it up. Once you dry it up, this stem grid is introduced into the electron microscope and we do the analysis. This is the very simple basics of working with the electron microscope. And here, this is another laboratory setting I am showing you. This is the lab of Indian Institute of Science, Bangalore. And you can see right side, there is an instrument sitting on the top of the table in front of the researcher who is sitting there. That was a field emission scanning electron microscope with energy dispersion spectroscopy. I was working with this lab for nearly 10 years from 2010 onwards. And here, the organic plant materials are studied in FESM, and metals and minerals and elements are studied in the HRTM, because HRTM produces high energy, and if you put organic material under the electron microscopy in HRTM, the material will get burned and these fumes will damage the aperture of the electron microscope. So nobody allows us to do this in the HRTM whenever there is a organic material in that. So I had to use 2 electron microscope in 2 different laboratories for almost 10 years and plus. And this is a team which helped me to work in the Bangalore Indian Institute of Science. And the man in the right side, you can see he was Girish Kunde. He was in charge of the lab. He was a scientist. And the man standing next to me is Varadaraja Perumal. He was a good friend of mine through the laboratory relationship for so many years. And they helped me to pull through the most difficult work of nanotechnological work in the lab, and the remaining staff there around us were all helpful. They were all part of the laboratory. And this picture was taken in 2015 after I completed my preliminary studies in the lab. So this is a basis I have to introduce you about homeopathy and how it is getting linked into the nanotechnology. It was an arduous task because whenever you go to a modern, advanced, fundamental laboratories, they are all basically skeptical about homeopathy because there is a lot of negative publicity all over the world against homeopathy for the last 200 years. So first thing we have to do in any research is overcome the social barriers, which we need a lot of willpower, and we can, with our willpower and abilities, we can pass over all that primary test. And because whenever we go to any lab, at least one person will ask you, why, doctor, you want to waste your time? Because homeopathy has nothing in it. And even homeopaths also used to say the same thing. Homeopathy has no material content. It is absolutely dynamic medicine. It is a spiritual medicine. And I have explained in detail how that part is dynamic philosophy came into homeopathy because of potentization. If you study about the Hanuman's life and works in detail, if you go through Richard Hale in detail, you will really understand Hanuman wanted to have a completely materialistic science. And that is what he envisioned for. In 1790, after the discovery of homeopathy, he said, yes, I found a material medicine which is perfect. But after 20 years, after the discovery of potentization, after 1810 to 12 period, Hanuman had to change his position. And as a scientist, he was willing to do that based on the evidence. At that point, Hanuman said, dilutions, potentization, trituration, and potentization leading to dynamization, dynamization leading to evoke the spiritual powers of the matter to cure the patient, because the science of that period was incompetent and underdeveloped, if you speak in the scientific terms, underdeveloped to establish anything in homeopathy. Because we have a way of thinking which is always linear in way. And in that linear fashion, later years, Avogadro's number became a very prominent theory in chemistry and physics. That was more than enough to debunk homeopathy as a placebo therapy. But my dear doctors, my dear friends, we are not holders of a placebo therapy. From the beginning, from the day one onwards, we were very clear that we are curing patients. and we cured millions and millions of patients around the world. These cures were not because of dynamic action of medicine, but it was the action of the nanoparticles and quantum dots in homeopathic ultra-high dilutions, and that regulated the human genetic system and the protein synthesis and the epigenetics even 100 years before or 150 years before we realize there is something called DNA, there is something called RNA, and we have identified all these things 200 years after the period of Hahnemann. It was not Hahnemann's fault. Definitely it was not homeopath's fault. And we had to take all the blame. Now we are debunking the science which is based on the material world and which always said that the science of the human beings are basically a material form. But we human beings are not only the material form. We have a mind which is not integrated into the treatment in any system other than homeopathy. There is only one system in this world which uses the both mental frame and the physical frame simultaneously to identify the drug and cure the patients. That is why homeopathy cures. So, my dear doctors, this research was extensive. You can see in the book Nanodynamics, there are more than 150 samples I have listed. Actually, I have studied more than 200 samples during the course of my studies. And in every drug, whether it is metal or mineral or plant or imponderables or no sorts, everything you could see that there are nanomaterials present. And how this nanomaterials comes if there is nothing in it. And I am coming to that point. We have also studied placebo, that means alcohol control samples diluted up to 30 potency in 2 different electron microscopes where I studied all the homeopathic dilution in the same electron microscopy and nothing we found. There was nothing. So my dear friends, we know the secret of homeopathy is nothing but potentization. This is the one point where Dr. Hahnemann made a tremendous stride from the grams and grain doses of medicine to the nanoscale of medicines, which he was unaware of that. And this product we call potencies, both centesimal and 50 millisimal scales. And potentization made homeopathy unique. Potentization made homeopathy individualistic. Potentization made homeopathy, I can say in today's science, the most advanced medicine in the world. So what is nanoscience? Nanoscience started evolving after 1960. Definitely it was there before that when during the period of Faraday itself, Michael Faraday itself, it started, but it took a definite shape after 1960 and it became evolved after 1990. So it is one of the most recent science in the, if you relate to physics. So 1 nanometer is 1 billionth of a meter. Or you can say 10 Armstrong is 1 nanometer. 1 Armstrong is 1 hydrogen atom. So 10 hydrogen atoms, if you put together, then you call it is a nanometer. You can understand the size when you go to the nanoscale is such a small. So what is the importance of electron microscopy? If I tried to do the same analysis 30 years ago, I would have failed because the electron microscopes didn't reach these specifications and this level of accuracy 30 years ago. And even 25 years ago, it was not there. And we need electron microscopy which connected with energy dispersion X-ray spectroscopy, which is not common in every lab. It is also very uncommon. But without a spectroscopy attached to the electron microscope, our studies will not reach anywhere. Under an electron microscope, we can identify the particle. But the content of particle cannot be identified if there is no spectroscopy attached to that. So that is a unique position. It is a, there is a time to come out anything. Now only the time for homeopathy has evolved. Now only the equipments or electron microscopy has developed to the level of where homeopathy can be identified. So another machine I used is dynamic light scattering, which is more common equipment used in the nanotechnology to identify the particles in a gross level. And this is what I'm going to show you only one picture here. That is on aurum metallicum. Aurum metallicum I studied from 6C to CM potencies and also LM1 to LM30 potencies, which you can see in the nanodynamics book. So you can see here, the particle size is 0.3 nanometer to 20 nanometer in the case of 6C potency in 6 hours of trituration. Why homeopathy and nanotechnology we are linking? I can tell you simple thing. We do trituration and succussion. You take aurum metallicum, we are doing 6 hours of trituration. As every homeopathic pharmacy student and every doctor is aware, any insoluble material and any metals and minerals, we are doing trituration. That is 1:99 ratio. And after 1 hour trituration, we go for 1% or one part of it with another 99 part of sugar of milk and go for second stage of trituration. Repeat it 6 times. That is called 6C potency. This is the 6C potency of aurum metallicum. And you can see the resolution power here 100 nanometer, 50 nanometer, 10 nanometer and 2 nanometer. And you can see the particle size is 30 to 20. And this is the HRTM image of particle size of 30C is 3 nanometer to around 10 nanometer. And you can see the resolution 200 nanometer, 100 nanometer, 50 nanometer, 20 nanometer. And in the 50 nanometer, you can see there is a pentagon-like structure. Within that, thousands of nanoparticles are embedded. When you go to nano world, it is amazing. And many things which will make you to realize the dimensions of a different world. Such a small space has a lot more than we can imagine. So this is aurum metallicum 200C. You can see there are in the 500 nanometer resolution, you can see the pictures resembling to the ornaments. That was beauty actually. When you go under electron microscope, many natural formations we will see. And in 100 nanometer resolution and in 20 nanometer and 10 nanometer resolution, 10 nanometer resolution picture is very, very unique. And you can see individual atoms of gold embedded in that particular substrate. That is a very rare thing which happens when the sample preparation and you're focusing, everything is perfect, you get it. And let me elaborate a little more. I have shown you a TEM grid in the earlier slide. And this TEM grid, I told you there are 200 columns. And pictures I am showing you here is one corner of one column. In one column, this resolution is 10 nanometer from 50 micron to 10 nanometer. And this 10 nanometer is what you are seeing this material here. So this is something very unique. And go to the images of aurum metallicum 1M. Particle size is slightly reduced. The basic particles all are less than 10 nanometer. These 50 nanometer particles are mostly coming as aggregates. That means many particles join together. They are not independent particles most of the time. Majority of particles of all the metals after 30C potency are within quantum dot scale, less than 10 nanometer. We call nanoparticle less than 100 nanometer. When they are between 1.5 to 10 nanometer, we call them quantum dots. And quantum dots have a totally different properties, physical, chemical, mechanical properties compared to the larger particles of the material. Look at the aurum metallicum 1M. In the 500 nanometer resolution, you are seeing a beautiful picture. And that picture resembles ornaments, gold ornaments. So we make ornaments, and in nanoscale, when you collect all the golden nanoparticles, they naturally himself make ornaments. So this is the beauty of the world, and this is the beauty of the nanoscale. This is the second sample that is 10 nanometer resolution is slightly burned because of the energy which passes through that substrate, burnt because there must be some amount of organic material, maybe carbon. Carbon is constantly present because we are preparing medicine in sugar of milk. So carbon will remain in most of the places. And 20 nanometer and 10 nanometer resolution you can see. This is orometallicum 10M potency. This is 25 to 50 nanometer. And here 10M potencies, when the potencies are more and more higher in metals, I have noticed particles become more subtle and they immediately get coagulated and come aggregated together. So getting independent particles sometimes is more difficult. You are getting a combination of particles in many occasions. This is the orometallicum 50M. And here also the smaller particles you can see 3 nanometer. And here 50M potency then followed by CM potency of orometallicum. Here the particle size is 7 to 100 nanometer within nanoscale, but within or quantum dot scale also particles remains. And in all these places, this is not only with aurum, this is with platina, this is with ferrum, this is with every drug I have studied. There is the original material of the metals and minerals in all the metallic and mineral drugs. And in organic, it is carbon and oxygen in different combination because organic materials are combinations of organic carbon, hydrogen, oxygen. So this is a table. You can see the percentage of carbon and aurum. Aurum you can see in the first column. Every potency has different percentage of aurum. And so many other materials are coming in between. And copper will show persistently because we are using copper wires in the 10 grid. So naturally that get reflexes. This is the percentage of aurum in different potencies. This is not an absolute reading because this is the reading of a very small area in the 10 grid, not the complete area in the 10 grid. We are unable to do that. So maybe further studies will lead to understand 1 ml of aurum metallicum 1M contains how many nanograms of gold we can identify in the future. So we can standardize the homeopathic medicines in 30 ml of aurum metallicum. What could be the content of aurum? How many nanoparticles are roughly there? How many million particles are there? What could be the nanogram or picogram of nanoparticles of aurum metallicum into that? That should be the next stage of study. All you youngsters can take up such a study for the future. That will absolutely standardize homeopathic dilutions and potencies, and we will know exactly what we are giving to the patient. So this is analysis of the control samples of aurum metallicum, not aurum metallicum, alcohol control samples in field emission scanning electron microscope and HRTM image, second one of the TC. For curiosity, I just put one more picture here, that is arsenic alb. And the size of nanoparticles of arsenic alb is 5 to 71 nanometer. And you see the morphology of the particles from aurum to arsenic, it is entirely different. And these particles of arsenic, why I put it here? During the COVID epidemic, our Ayush Ministry initially said arsenicum 230 can be given as a preventive or a curative for arsenic or for the COVID fever. Suddenly people started attacking all the social media with anti-homeopaths have become very active. They said there is nothing in it. And some people will say there is nothing in it, and other group of people will say arsenic is a poison. The same time they say there is nothing, it is a placebo, and another tongue will say it is a poison. But the thing is, in the arsenic 200C, you have arsenic nanoparticles, but they are in nanoscale and in quantum scale. And they get converted into arsenic dioxide. Arsenic we are using itself is arsenic trioxide. And arsenic must be converted into, not only for arsenic, I have studied in platina, I have studied in ferrum, everywhere I could see that these materials, especially in platina, I did a specific study of selected area electron diffraction, SAED. And all potencies of platina, the nanoparticles are not platinum, they are platinum dioxide. So when they get oxidized, their toxicity will go away. So arsenicum, oxidized arsenicum, when you use in a nanoscale, it never produces toxicity. So this is something scientifically very clear. We are using almost all poisons in this world, almost all heavy metals in the world. and we are curing the patient. If you are using 3x or 3c or 1c, there is a possibility you will damage the patient because that particles can damage the organs, especially kidney or liver or many other organs. But when you use in nanoscale, a chemical reaction, a toxic reaction is possibly very rare. So what is my conclusion of study on this? We have every homeopathic dilutions of aurum metallicum as well as other drugs have nanoparticles and quantum dot sized particles. And the basic metal, basic element, basic material, that is aurum in this case, is present in all potencies of aurum metallicum. This is what I have seen in every drug. So this is one aspect of the study. Regarding when you come to the advanced therapeutics of homeopathy, we need to understand one thing, which is not well understood by the medical profession even today. Because usually we say that biological systems always travel 50 years behind the physics, minimum. So physics has moved from the fundamentals from atoms to subatomic level. But biological medicine, especially the modern medicine, is still standing on the dualistic principles of Descartes, that is Cartesian dualism. That is, mind and body are separate entities because they have no idea how to do with mind. So they just keep the mind away from the table and say, look at the body. Body is made up of organs. Look at the organs. Organs are made up of tissues and cells. And tissues and cells and all these things are chemically modulated. That is why chemical industry is the basis of conventional modern medicine. They never had an idea about the DNA, RNA, anything when the discovery of antibiotics happened. Discovery of antibiotics happened in 1929. Commercial production of antibiotics happened in 1944 March. DNA was identified in 1953 and Watson and Crick. And you saw in the newspaper yesterday, one of the great scientists in history, he was part of the discovery of DNA in structure of DNA in 1953. Watson, I suppose, if I'm sorry, if I'm wrong, I'm sorry, died yesterday at the age of 97. In the newspaper, it was a big news. So, my dear doctors, we are all studied anatomy, physiology, biochemistry, pathology, everything. Modern medicine based on two tenets, that is Virchow's cellular pathology, pathological basis of disease on one side, and another side is microbiological basis of disease. These are the two pillars on which the conventional modern medicine stands. They have no material in their hand to look at a epigenetic or a gene regulatory mechanism. But biological system, how they are operating. The first person who identified that the human biological systems are nanoscale machines, biological nanoscale machines, was Richard Feynman. He was a Nobel Prize winning physicist. In 1960, during the American Physical Society conference, he delivered a speech and he said, if you want to create nanoscale equipments, look at human body. And he said, everything in the human body is operating in nanometer scale. That time, no biologist realized what is going on. Now we realize that our DNA is a nanostructure, our RNA is a nanostructure. Every hormone, enzyme, receptors, antigen, antibodies, much more than that. Even the cells in the liver, Kupffer cells, etc., are all nano devices. Look at the hemoglobin, which is carried in the red blood cell. It's a nano device. White blood cell, it's a nano device. So biological nano devices are the operating principles in the human body, controlled by the gene regulatory mechanism. This is the core of human existence, which is nothing to do with conventional modern medicine, but which has everything to do with homeopathic therapeutics. That is why we are coming into the modern medicine. The modern medicine, if you are seriously looking with a scientific point of view, it should be renamed. Homeopathy should be renamed because this is the most modern therapeutic way to reach the inner self of human being, inside the cell of human being, inside the nucleus of the cell, and make regulatory changes in the genetics that we call epigenetics. So the genetic basis of biological existence is the core of human existence. So you look at a human being, start the journey from a single cell in the mother's uterus after the combination of sperm and ovum. And this gene regulation is the basis of growth, existence, health, disease, even death. So current mechanistic model of conventional biomolecular medicine is absolutely an outdated model. It is absolutely outdated in scientific terms. It has nothing to do with gene regulation. It has nothing to do with the controlling system in the body. It is only doing chemical regulation in the intracellular, more than intracellular, extracellular fluids. And physiological changes, physiological corrections, physiological manipulation, biochemical manipulation. This is what does the conventional modern medicine. And that is why they are a business model. Once you become a patient, you take lifelong medicine to regulate the body system in a physiological and biochemical level, never cure a patient. We homeopaths, instead of that, we are not curing everybody coming to us. Nobody can do that. But we cure a good number of patients forever. They will completely get rid of their sickness. You take an allergy, you take any disease, for example, a skin condition, or you take anything like migraine, a headache, where people are on years together on painkillers and other drugs, are completely getting rid of everything after a couple of months of homeopathic medication. How it is happening? Is it going to happen with a placebo? No, it's never possible. It is possible only there is a subtle science in that. That subtle science is homeopathy and its therapeutic principle and its material in a subtle nano level. That is the pharmacodynamics of homeopathy. So if you look at the human body in a little more deeper way, the single cell which we have in our first inception become 80 to 100 trillion cells in the human body for different functions and different performance and different structures. But what creates it? It created by the DNA which is there within the cell nucleus, which is 2 meters in length in every cell. So if you take 100 billion cells, trillion cells, and count 2 meters on each, our body DNA has a length of nearly 20 billion kilometers. That is too big. That is double the size of solar system. Such a complex system is operating in our body. And even in that, we have a coding part of DNA. We know adenine, thymine, guanine, cytosine, and the DNA coding system. And that coding system is only 2% of the DNA. 98% of the part of DNA, which is not coding, it is non-coding part, is still unknown to us. What are the major functions? Now we realize that they are acting like switches to switch on and switch off the coding part. So this is very, very interesting. If you look at modern science in the terms of homeopathic perspective. So epigenetics and nanopharmacology, we know that homeopathic ultra-high dilutions contains nanoparticles of the drug materials. And these nanoparticles are always capable of gene regulation and epigenetic modifications. And that is why in drug proving, as homeopathic students, I am sure all of us have done at least proving of one drug. If we didn't do that, I request you, every one of you, to do the proving of at least 5 drugs in your lifetime. And during your course of BHMS, at least 2 drugs you have to prove. When you are a postgraduate student, another 5 drugs you have to prove. Anything new or anything already proved. I can assure you, you are going to be the best ever doctor in the world because you will be confident about what you are doing. When you start taking medicine in a drug proving, it produces signs and symptoms. When you take placebo, you will never produce signs and symptoms. And if you have 50 provers in the group, everyone produces some symptoms very common. How it is happening. So these symptoms are what we are taken, signs and symptoms are what we are taken, and the totality which is taken to cure the patient. Therefore, the concept of similia similibus principle came. So pharmacology and pharmacodynamics is the basis of homeopathy. And the philosophy is a rational explanation given to homeopathy when the science was not very clear. When the science is more clear, less philosophy involves. But without a philosophical basis, there is no science can survive. The philosophical conflict is what killing allopathy today. But the philosophical comprehension is what making homeopathy great today. So, my dear friends, things are very different when you go to the nucleus where the DNA operates. From the DNA, RNA takes the commands and the transcription happens, and RNA splicing and RNA transport and translation of messenger RNA into the ribosomes for protein synthesis. This is the core of human existence. And this is where homeopathy operates. This is my own creation. You can see the things in a different way here. Look at this slide very carefully. We have the DNA in the genes, RNA formation, protein synthesis, then it come out of the nucleus into the target organs and make the cell structure, cell function, enzymes, functions, the hormones, and everything. And here, the basic level is DNA and RNA and protein synthesis. This is where in the epigenetics, homeopathy works. That is where the nanopharmacology of homeopathy works. Whereas so-called modern molecular biomedicine works in cellular pathology based on cellular pathology and microbiology is on the cell, enzyme, cell structure, and also the biochemistry of the fluids. This makes huge difference. I can assure you homeopathy is 100 times, 1,000 times scientifically advanced than conventional medicine. But we need to fine-tune this process. We are still operating with a lot of hypotheses, trial and error. If you do a lot of scientific experiments, I'm assuring you in another 1 decade is going to be critical for the history of medicine, where the homeopathy can cure the patients individually on the basis of individual genetic peculiarities. This is going to be the true sense of personalized medicine, which others can never think of. So this is what we call gene expression. The entire process from transcription to the translation of RNA code and the formation of proteins in the cell cytoplasm, all together we call gene expression. And this is where homeopathy operates. So homeopathy is a nanopharmacology. And the importance is, you look at the pharmacology of conventional medicine. Most of them are operating in the biochemistry of the body. None of them will go and operate intranuclear compartment. Any conventional medicine drug operate in intranuclear compartment is generally toxic. We have seen it many times. But because their medicines are based on the quantity, based on the weight per kilogram, that means how many milligrams or how many grams per kilogram body weight is the standard formulation in the pharmacodynamics of conventional medicine. But homeopaths, when we are giving 5 pills or 1 pill or a quarter drop of medicine to the tongue of a patient, we are not giving medicines in milligram or gram scale. We are giving medicines in a nanoscale or quantum scale. And they are having free entry into the cell cytoplasmic wall and cell nucleus wall. They can direct entry into the DNA and the specific genes they can activate. This is the epigenetic programming of homeopathy. So they also act on RNA, hormones, enzymes, receptors, antigens, antibodies at the nanoscale. Problem is, human body is composed of nanobiological devices. And for biological devices to act something on that, you need still more smaller medicinal substances. Homeopathic medicinal substances are basically still more smaller than any nanomaterials in the body. I have shown you the picture of arsenicum album 200C earlier, and the size of particle begins from 5 nanometers somewhere. You know the size of the virus. COVID virus, the minimum size was 135 nanometer. And a homeopathic arsenicum album, a nanoparticle is 3 to 5 nanometer in size. They can operate even on viruses directly, not only promoting the human immune system. So this is amazing. What is in your hand, in the hands of homeopaths, is unbelievably subtle. And you can do magics with that. And we are doing that. But we should be confident to tell what we are doing, confident to understand what we are doing, and we should be strong enough to defend what we are doing. So I would like to call Hahnemann as the pioneer practitioner and founder of the concept of nanomedicine. And definitely Michael Faraday is considered as the father of nanoscience. So this is where you can see the comparison. I am coming to the philosophy of homeopathy in the next weeks, and there I will compare the philosophy of conventional modern medicine and philosophy of homeopathy, and there these points may come back again. So the modern medicine which we are seeing today, people say it is a scientific discoveries are happening every day. It is all like a Maya which they are distributing everything. The whole basic concept of modern medicine got crystallized with the cellular pathology of Virchow in 1858 and molecular theory of Robert Koch and Louis Pasteur before this century, of previous century. And the current structure of conventional modern medicine was not created by any scientist or researcher. It was created by a man called Abraham Flexner. He was a Johns Hopkins graduate and a young boy around 20s, 20 to 25-year-old guy. He was a commission from 1905 to 1910 instituted by John Rockefeller under the John Rockefeller Foundation, paid him $2 million to go around US and Canada and study about the health system and how to promote the pharmacological department of Rockefeller Foundation, John Rockefeller. That company was called, now also we know that during COVID time, we have got COVID vaccines, and they had allopathic pharma industry with them. And they found there is not enough sales for their medicines. They sent this fellow, John Rockefeller, to study about it. And John Rockefeller gave a report to Abraham Flexner gave a report to John Rockefeller in 1910, and later by 1920 it became the basic report of health education in America, because some of the American presidents approved this report as an official document. So the capital power of John Rockefeller is the basis of current today modern medicine. And Second World War happened and antibiotics became a boom, and antibiotics saved millions and millions of lives during Second World War. So it became unquestionable. And homeopathy, which was number one in the US, got completely wiped out almost. Still it is there. There are very good practitioners, doctors, but it is in the back end. So this is all meant for contain infections and injuries for an emergency. It is an accidental emergency and infectious medicine, not a disease treatment for chronic diseases. That's what proved in the last 100 years. That is where the conventional medicine is failing tremendously. So they are dualistic. Why they are dualistic? If you have a mental symptom along with a physical problem, for the mental symptom, you should go and consult a psychiatrist. If you look in that angle, a homeopathy is the only comprehensive holistic physician in the world. A patient comes with an acute infection or a chronic disease, severe anxiety. You don't need any psychiatrist to treat that. You don't need any clinical psychologist to counsel him. You act like a counselor, you are becoming a psychiatrist. You give arsenicum album, or you give orometallicum, or you give argentonatricum, which covers his physical ailment as well as mental symptom, and he will get cured for both. Which system of medicine can offer such a comprehensive treatment strategy other than homeopathy? Only homeopathy can offer it. And we are doing and we are curing patients like that. So modern medicine failed to cure all chronic diseases. I can say all chronic diseases, but it succeeded in managing all emergency situations, life-saving, as well as infections in the last 100 years. And the medicinal, the involvement of advanced technology and the science helped the modern medicine to perfect the art of emergency medicine. And that is where the modern medicine is strong. Whereas if you look at the therapy and treatment of complex genetic involved diseases, it is a big zero in front of homeopathy. So nanoscale medicinal agents act in homeopathy not as an antidiabetic, not as an antihypertensive, not as an anti-allergic or anti-tumor or anything in milligram or gram doses, but it acts in a nanoscale, in a nanograms, and there is what called the nanopharmacology of homeopathy, which cures the patient, permanently cure the patient. So the reality is the scientific world is evolved in the last 500 years or last 2500 years, started from Aristotle in the West. Then the next turning point came with Newton's law of physics. Then after 400 years, the next change happened with Einstein and Rutherford and Max Planck and other scientists in physics. And we are landed from gross matter to subatomic particles in physics. But in medicine, we are still in the gram and milligram and kilogram doses of medicine. But this is going to change, I can assure you. We are sitting on a volcano of change. So biological existence of human beings are defined by Cartesian mechanistic model. Descartes is the first philosopher in 16th century defined that body, human organism or human existence can be divided, or human body is combination of machines. He said machines. And human mind is independently existing, not in relation to this machine. So he made the human existence into two, the mind as a separate and the body as a separate one. And what he believed is like a machinery, if all organs and systems are perfect, human body become a perfect machine. This mechanistic model is what is devised for the basis of modern medicine. Still they are struggling how to comprehend this duality. This is not going to work when the genetics is coming into the prominence. This system is going to fail. So DNA coding is the basis of growth, health, disease, as well as death of any biological organism. And that will teach us one thing very precisely. Chemically intensive, biochemically and physiologically dependent, conventional therapeutic method is already failed. It is on the deathbed. And we are not perfect at our art and science. That is why world is not having a proper therapeutic method. We need to do that. That is what we are doing in the last 10-15 years. And that is what the profession needs to take up and take forward for the future. The homeopathic potentization retains nanoparticles in all dilutions. So Avogadro's principle is not eligible or not capable or not qualified to assess homeopathic dilutions. Homeopathy is a purely personalized therapy because it is a purely, truly personalized nanomedicine. Why? The reason is homeopathic individualization of patient is a way to sketch the genetic blueprint of the person using individual expressions of signs and symptoms. Why I am so sure about it? Individual expressions in a patient in any characteristic symptom we are taking up or any common symptom has based on a specific gene regulatory change. This is a pure science. If a symptom is because of specific gene regulatory change, and if you are curing a drug, a patient with a specific symptom, with a specific drug, which produces similar symptoms during drug proving, what are you doing? You are touching the specific gene and re-regulate it. So the disease is cured. Every expression of a human being, every expression, every symptom, every sign of a human being has its root in the DNA coding. So epigenetics is the basis of treatment. Epigenetics is the basis of homeopathy. But the pharmacology we are using is not the chemical-based pharmacology. We are using quantum-based and nano-based pharmacology. This is an in-depth science I would like to talk with you today. So we are post-COVID, people are understanding much better new sciences, new ideas, and the total failure of conventional standardized statistical medication to the world, which was an absolute failure. I have written 7 articles during COVID time, especially in 2020-21 period before the arrival of the COVID vaccines, that the whole management of COVID is wrong and the vaccine is going to do more harm than the total COVID did to us. This is what we personally, all of us experiencing today, because vaccine injury is killing thousands and thousands and making incapable millions of people around the world. And nobody takes responsibility for that. That is a criminal offense. That was really a crime against humanity. So this is what I would like to say today. My dear students, my dear friends, my dear doctors. We are not seeing each other, but I can visualize all of you in front of me. Homeopathy is a blessing. I always felt like that the day I joined for homeopathy. I don't know why, but for me it was like that. I had no tradition of homeopathy with me. I am the first person in my tradition and family to join homeopathy. But after 1 year, I realized this science is going to be the science of future. I started working, I started practicing, I started teaching, I started researching, and the results are here in front of all of you. And these results are definitely going to make huge change in the world in the years to come. So homeopathy is a pure science. It is a material science at a subtle nano quantum level, and homeopathy operates in chain regulatory mechanism, and homeopathy cures the patient. And what I believe strongly, with the advent of artificial intelligence, more and more knowledge about genetics and information technology together can make homeopathy a perfect science in the years to come. And you have to be there to take the fruits and to eat the fruits and carry the lamp of homeopathy to the next generations. And these are the important research papers I would like to share with you. And these research papers are, most of them are freely available, and some of them may be paid one. And you have to download and read this. And I requested all colleges where I contacted to tell the first BHMS most junior students to attend the program, because this is the basic science of homeopathy, which you may not get exposed in the academic curriculum, because curriculum makers are ignorant about the modern sciences. And none of them were researchers, many of them were not even teachers. So they make curriculum and syllabus without knowing how the world is evolving. They look at their personal priorities and create syllabus and curriculum and textbooks based on the interests of 1950s or 60s or 70s or 80s. But you are not going to live in the 20th century. You people are going to live in the 21st century. So let us be the masters of ourselves, and our knowledge is not to be dictated by some of the ignorant people in the world. So, my dear students, these are some of the important research papers. Nanomaterial characterization of diluted platinum and alcohol control sample. In this paper, I have come out with a very, very new discovery, which I told you earlier, that metals, especially the noble metals, when you potentize, they become oxidized. We call them noble metals because they never get oxidized. But in the homeopathic process of potentization, they are getting oxidized and they are creating wonderful cures in the human body. Homeopathy as a personalized nanomedicine. This is also another paper written in the journal Homeopathy, and many other papers written in this is an American Journal of Homeopathy, and many other international journal of high dilution research. And I debunked the Avogadro's claim of Avogadro's number by scientists that homeopathy is unscientific and there is no material as per Avogadro's number. The whole Avogadro's number is a sham as far as homeopathy is concerned. So we reject it. Absolutely, we reject it. And the many other papers I have published on fundamental research, especially related to electron microscopy, spectroscopy, and you can read these papers, make yourself relevant in the years to come. You need to be relevant. If you need to be relevant, you need to be updated scientifically, politically, socially, also economically. So that is very, very important. This is another important book I have published in 2019, Ray of Civilization, which threadbare look into the nuances of medical science at some point. And this is more than that. This book is much more than that. You can read, it's available in Amazon, you can buy and read, and that will make you much better as a physician. And these are the other two books which are going to be explained in the next weeks, 18th and 20th. 18th, we are going to talk about the nucleus lectures on chronic diseases and miasms. And 20th, we are going to talk about lectures on homeopathy and philosophy, that is New Lights. And these are the books translated into, this is the first one is a Russian translation of Nucleus. Second is a Turkish translation of Nucleus. And I have shown the other translations earlier, the German translation of Nanodynamics. So my dear doctors, friends, young doctors and students, I can tell you one thing. If you go deep into homeopathy, you will have a unbelievable, extraordinary time ahead of you. You will control the world. Thank you so much. Thank you for time. Thank you for your listening. Keep in touch. Anything for everything related to this. Thank you, everyone. Thank you, Dr. Noori, for your chairmanship. Thank you.
Thank you so much, sir, for that absolutely fascinating and deeply insightful presentation. The way you presented the scientific concepts with such clarity and practical relevance has left us all inspired. I'm very certain that the listeners have gained valuable guidance and a new perspective today. And there are a few queries. If you allow, we'll take them.
Yeah, absolutely. No problem.
Uh, So let's take this question from Anvesh. He's asking, according to you, if you are a scientist and a layman asks how homeopathy is working, what will be your answer?
That is what I was telling all this time, how homeopathy is working. It's a nano medicine and a quantum medicine. It's operating in gene regulatory mechanism and epigenetics. That is what we are identifying now. Thank you.
Dr. Y.N. Rao is asking, are homeopathic medicine and nanomedicine synonyms?
No, it is not synonym because modern medicine using epi drugs, they call epi drugs, and they're trying to invent epi drugs, and they wanted to, the concept of nanomedicine in conventional modern medicine is very superficial. It is 99% of research is going for utilizing nanoparticles. If you look at nanodynamics book, you can show, I have given the type of nanoparticle. There are branched nanoparticles, a lot of nanoparticles, rods are there, so wired nanoparticles, so many nanoparticles are there. What is the research going on in conventional medicine is transport of chemical molecules of chemotherapy from to the target organs by using nanoparticles. That is what they call nanomedicine. But for us, nanomedicine is our medicine is nanomedicine. That is the difference. Clear?
Dr. Srinivas is asking, where are the OH particles in Auramet nanoparticles photo and how do they look at such magnifications?
That you can feel if you are going and sitting in front of an electron microscope, which cannot be explained actually. I was wonderstruck when I was studying about Natremur LM24. I was studying Natremur LM24 under HR-TEM. And at a particular moment, myself and the technician were awestruck because particles started dancing. Because when the electrons are passing over it, they absorb the particles and this energy they utilize and they start jumping. So under electron microscope, you are seriously sitting, you will find very, very interesting things.
Yes, I'm
That
sure.
is of the subtle world.
Dr. Anvesh is asking, any latest things you have understood and found in these 2-3 years of your latest works?
Actually, I'm working and definitely in the next year, 2026, you can expect me to come out with a very different, entirely different perspective. Nothing contradicting what I said today, but a complementary to what I said today. The drug action in homeopathy will be having more clarity. I'm working on that for the last 7-8 years right now.
So your work truly takes us to the very heart of modern exploration, scientific exploration within homeopathy, sir.
100%, I can assure you.
As we come close to this remarkable session, I am going to express, on behalf of Homeopathy 360 as well as BGen, our deepest gratitude to Professor E.S. Rajendran for sharing his invaluable research and vision with us. To our audience, thank you for your active participation and thank you for being part of this very intellectual exploration. Together, let us continue to advance the dialogue between traditional and technology, between heart, healing art, and molecular science. Once again, thank you so much, sir.
Thank you so much. Thank you, everyone.
Goodbye. Good night,
Bye.
everyone.
Bye. Bye. Bye.